Well, welcome everyone to this podcast for Biocore 383 on the TCA cycle and reducing power. One of the main goals of this podcast is to go through some of the detailed reactions of the Krebs cycle. Much of this is in your book, but it's probably nice to have a quick overview. Before we get into the details, however, it's probably worth reviewing some of the goals that we have for this unit in 383. As well as discussing where these reactions occur. So let's start with the goals. I want to remind you that we want to know several things about the Krebs cycle. First of all, what are the inputs and outputs? What molecules enters the cycle? And what are the outputs of the cycle? In particular, we want to know what are the chemical goals of the cycle. And just as a reminder, the main chemical goal is to produce reducing power that we will use via electron transport during oxidative phosphorylation. As always, we want to remember where these reactions occur. Location is really important for biochemical reactions, and that is most certainly true of these reactions, as we'll see. And then finally, what we want to address is how does this cycle feed into high efficiency ATP production? Given the chemical goals of the cycle, how does that relate to the actual production of ATP that occurs during oxidative phosphorylation? Now, again, our goal here, though, is to delve into a bit of the detail of the cycle. And you'll notice that sometimes I use the words TCA and Krebs cycle interchangeably. Actually, there's a third way to describe the cycle. It's called the citric acid cycle. The name Krebs comes from Hans Krebs. Here's Hans. And Hans was the one who realized that the reactions of this cycle, in fact, are cyclical. A lot of people had tried to figure out these reactions, and they had assumed that they were linear, kind of like glycolysis is. Krebs realized you couldn't make sense of the data in that way, and he had the brilliant insight that we actually regenerate components of this cycle once we go once around the cycle. Right, so where do these reactions occur? Well, remember that glycolysis occurs in the cytosol, the peach region on the left in this diagram, which is figure 10.1 from your text. And there, we took a 6-carbon molecule, glucose, split it in half to form 2 pyruvate molecules. We got a little ATP out of that, and we also got a tiny bit of reducing power in the form of NADH. The TCA cycle occurs within the interior of the mitochondria. And in particular, most of the reactions of the cycle occur in the matrix of the mitochondria, in that dark brown region on the right in this diagram. So that means that we have to get pyruvate into the interior of the mitochondria where we can work with it. Pyruvate is produced via glycolysis in the cytosol, and there is a facilitated transporter in the outer mitochondrial membrane, the dark orange region in this diagram, called a porin. And porins allow pyruvate then to go through the outer mitochondrial membrane. In eukaryotes, however, ATP is actually expended to get pyruvate through the mitochondrial inner membrane. This occurs via active transport. So here again, we're actually using energy to get the pyruvate in, but there's a big payoff as we process the pyruvate and extract all of the energy that we can out of it in the Krebs cycle and ultimately through oxidative phosphorylation. The NADH that's produced during glycolysis actually can't go through the membrane either, and so there are different ways of shuttling the electrons that are carried by the reduced form of NAD plus, NADH, across both membranes to get them into the interior of the mitochondrion where they can participate in electron transport. Because the amount of energy required to do the shuttling depends on the shuttle that's used, and that depends on the cell type that we're considering. I'm not really going to talk about that at this point, but if we do the total ATP accounting in terms of how much ATP we can derive from a single glucose molecule, that number will actually vary depending on which shuttle is used in any particular cell type's mitochondria. Okay, so with that preface then, let's consider the steps of the TCA cycle. You can see in this figure, which is figure 10.6 from your text, we've numbered these key phases in 6 steps. The first phase at the top involves pyruvate itself. Once pyruvate is into the interior of the mitochondria, we transfer 2 of the carbons from pyruvate to a molecule called coenzyme A to create a molecule called acetyl coenzyme A. In the process, we remove a carboxyl group, which is given off in the form of gaseous carbon dioxide, the brown circle in this diagram. At the same time, we create a molecule of NADH, reducing power. The next reaction takes that acetyl CoA that we brought in and is going to use the 2 carbons that have been transferred to coenzyme A as an acetyl group and join them to a 4-carbon compound, oxaloacetate. Ultimately, that's going to create a 6-carbon compound, citric acid, or citrate, which is going to move around the cycle. We're going to process it in several ways to extract reducing power, a molecule of ATP, and ultimately we're going to create a 4-carbon compound from which we're going to extract additional electrons, and ultimately we're going to regenerate that 4-carbon compound at the top of the cycle, oxaloacetate. Now, to do all of this processing, we're going to do 2 more oxidative decarboxylations. That's the process whereby electrons are transferred to NADH and we liberate a carboxyl group as gaseous CO2. We're going to generate ATP, and that's going to involve a kind of a funky method in which we're going to actually create a molecule of something called GTP, which is related to ATP, and then we will actually create a molecule of ATP. And then we're going to take the resulting 4-carbon compound, succinic acid or succinate. We're going to do 2 more oxidations, and those oxidations are going to transfer electrons to 2 electron carriers, one called FADH2 and another molecule of NADH in step 6. First then, let's consider the first 3 steps in this process. Once pyruvate enters the mitochondrion, we're going to transfer 2 of its carbons to a carrier molecule called coenzyme A. You can see those 2 carbons from pyruvic acid are marked in the kind of the peach color on the upper left in this diagram, and you can follow those 2 carbons throughout the Krebs cycle in the detailed diagrams that follow in the rest of this podcast. In addition, this diagram, figure 10.8, shows in dark brown the carboxyl groups that ultimately are destined for oxidative decarboxylation and hence liberation of CO2. So you can see that the carboxyl group from pyruvic acid is going to be liberated in this first step of glycolysis, and that's going to create a molecule of NADH. The enzyme that catalyzes this reaction, pyruvate dehydrogenase, is heavily regulated. In fact, that reaction is essentially irreversible under physiological conditions, a very highly negative delta G prime for that reaction. And so it makes sense that cells would tightly regulate entry into the cycle at all at this point. All right, another thing to point out about this diagram is you can see that that little box there, it says from beta oxidation. Well, that's a process whereby carbons coming in from fatty acid chains can enter the Krebs cycle at the acetyl CoA step, so step 2. And we'll come back to that at the very end because this is one of the ways that metabolites can enter metabolic pathways. All right, so let's consider in a little bit more detail this carrier molecule, coenzyme A. So you can see coenzyme A schematically here. This is figure 10.7 from your text. So there's an adenine and a ribose on the bottom here, and then there are a couple of phosphates that bridge to a functional group called pantothenic acid. Pantothenic acid is a vitamin, so you get that from your dietary intake, and you can see that it's a very important component here in metabolic processes. Then at the top of the molecule, above the red boxed region, is a sulfhydryl group shown in blue. That sulfhydryl group is the place where the 2 carbons in the form of acetate are transferred onto coenzyme A to make acetyl coenzyme A. So So the acetyl group coming from pyruvic acid gets transferred there, and the resulting bond creates something called a thioester group between the sulfur and the acetyl group. And you can see that on the upper right in this diagram. So that's acetyl CoA. So we have this huge apparatus for basically carrying these two carbons from pyruvic acid into the Krebs cycle. Right, so far we've taken a pyruvate, we've made one molecule of NADH, we've liberated a CO2, and we transferred the remaining two carbons to coenzyme A to create acetyl coenzyme A. Those two carbons then are transferred onto citric acid to form citrate. And now let's go on and consider these oxidative decarboxylations that follow on the formation of citrate. So those are on the lower right in this diagram again, which is figure 10.6 from your textbook. So there's an isomerization reaction that involves citric acid to make isocitrate. And you can see this coloring convention. Remember, there are two carbons that are colored. Those are the ones that came in from pyruvic acid in the peach color. And then the dark brown carboxyl groups are going to be liberated as CO2 via oxidative decarboxylations. And you can see how this works. First, the carboxyl group on the upper right of isocitrate is liberated. We get a molecule of NADH out of that, and we liberate a carbon dioxide, as you see in step 4. And that leaves us with a molecule called alpha ketoglutarate. Remember, you're not responsible for all the detailed structures in the Krebs cycle, but for completeness, we're mentioning them here. Right, you can see that there's a remaining carboxyl group in dark brown, an alpha ketoglutarate, and an oxidative decarboxylation occurs again. This creates another molecule of NADH and liberates that second CO2. The result is the transfer of the remainder of the molecule that's left after this oxidative decarboxylation onto another molecule of coenzyme A. And this actually creates something called succinyl coenzyme A. So this is something called succinic acid coupled to coenzyme A. That's what's on the lower left in this diagram from figure 10.8 in your text. All right, so at this point, what we have done is we've taken pyruvate, we've transferred electrons to NADH and liberated CO2. This transfers the remaining two carbons to acetyl CoA. Those are then glued effectively onto a 4-carbon compound, oxaloacetate, to create citric acid. That citric acid then is, some of the bonds are rearranged. There are two oxidative decarboxylations that liberate CO2 twice and generate 2 molecules of NADH. And ultimately now we transferred the remaining 4 carbons to coenzyme A to make succinyl coenzyme A. Now let's go on and look at step 5 at the very bottom of this diagram. This is the one place in the cycle where we actually get a little bit of ATP out. So we take the succinyl coenzyme A, as you see in the lower right in this diagram, and we're going to take that molecule and transfer that thioester bond away from acetyl coenzyme A to liberate succinic acid. Now in the process we can generate a molecule which is similar to ATP called GTP. And then there's a second reaction that takes the phosphate from GTP and transfers that phosphate onto an ADP molecule to ultimately generate a molecule of ATP at the lower left in this diagram. So we do get a tiny bit of ATP, but if you've been following along, this is actually the only place in the entire cycle where we generate ATP. So clearly the main chemical goal of the Krebs cycle can't be to make ATP. Instead, it's to make reduced compounds that are going to be used ultimately via electron transport to generate ATP through something called ATP synthase. All right, so we're making progress. Let's go back to our zoomed out view of the Krebs cycle. At this point, we have a 4-carbon molecule called succinic acid or succinate, And now we're going to do 2 additional oxidations, which will extract as many electrons as we can from this entire process. Those 2 oxidations are going to transfer electrons onto a slightly different electron acceptor called FAD to create FADH2, and then a second oxidation of the remaining molecule is going to transfer electrons onto another molecule of NAD+ to generate yet another molecule of NADH. And here are those reactions. So you can see that we take succinate, and we are going to oxidize it once. This is going to generate something called fumaric acid or fumarate. Then we're going to add a hydroxyl group to that to make malic acid or malate. And then finally we're going to oxidize that to generate another reduced compound, NADH, up at the top. And when we're all done with that, we're actually going to regenerate oxaloacetate. And so a 4-carbon oxaloacetate molecule can re-enter the cycle. We can add 2 carbons from a fresh copy, if you will, of acetyl coenzyme A to generate a 6-carbon molecule that's going to go through another round of the Krebs cycle. All right, so again, we're going to generate 2 more reduced compounds. FAD is reduced to FADH2, and we get this other another copy of NADH produced through this process. So it's worth looking at the structure of FAD. Here it is, and it's similar to NAD+ and NADH. Here we have FAD on the left. You can see that its structure is not too dissimilar from, at least the bottom part of it, from what we saw for NAD+. And we have another functional group, and this functional group can undergo oxidation or reduction, just as we saw with NAD+. This functional group 2 actually is provided in our diets as a vitamin. It's called riboflavin. So you can see at the top then that we can add hydrogens to reduce the riboflavin, and when we do that, we add 2 hydrogens, we get FADH2 on the right. So this is similar to NADH. It's a reduced compound that's an electron carrier. All right, so we've done a lot here. At the risk of being overly repetitive, let's review what we've done. All right, first, we took pyruvate into the mitochondrion. We undergo an oxidative decarboxylation. This transfers electrons to NADH, and this liberates CO2. We take the remaining 2 carbons and we transfer those to coenzyme A to create acetyl coenzyme A. And this brings in 2 carbons into the Krebs cycle. These are transferred to oxaloacetate to create citric acid or citrate. We undergo 2 more oxidative decarboxylations at that point. This liberates the remainder of the carbon dioxide that's liberated in the Krebs cycle and creates more NADH. There's one step in the cycle where, via a GTP intermediate, we make ATP. That's step 5. And this creates a 4-carbon compound, succinic acid, which undergoes 2 oxidations, liberating FADH2, an electron carrier, and NADH. And ultimately, we regenerate oxaloacetate. It's really an elegant process when you stop to think about it. All right, so the last thing we want to consider here is, as we did for glycolysis, where do metabolites that we get through our food fit into this process? Well, one of those metabolites are fats. Triglycerides are fats. And triglycerides can be broken down, and we saw that the glycerol from the triglycerides actually enters into glycolysis. Well, the fatty acid tails enter here via acetyl coenzyme A by a process which I won't describe, but it's described nicely in your book if you're interested, called beta oxidation. So fatty acids feed into metabolism by acetyl coenzyme A and the Krebs cycle. Another set of important metabolites we get through our diet are amino acids, and they also feed in at this level at the Krebs cycle. And you can see that here. This is figure 1012 from your text. And what this shows is the pathways whereby several amino acids feed into various components of the Krebs cycle. So alanine, for example, can be converted to pyruvate, and you can see where that would enter into the Krebs cycle. But other amino acids can be converted into intermediates in the Krebs cycle. So aspartic acid, for example, can be converted to oxaloacetate. Glutamate can be converted to alpha-ketoglutarate, a component of the Krebs cycle. So you get the idea. So this is similar to what we saw with glycolysis. So understanding these key pathways provides us with the entry points of things that we get through our diet into metabolic pathways. All right, so we've covered a lot of ground here. We've understood the entry points into the Krebs cycle. We've talked about all of the reactions of the Krebs cycle. We've talked about how metabolites feed into various components of the Krebs cycle. And this leaves us with a key question we haven't addressed, but we'll need to. First of all, what do we do with all this reducing power? We generated a lot of NADH, some FADH2. What do we do with that? And in fact, how are all these electrons that we transferred onto these reduced compounds coupled in some specific sense to efficient ATP generation? Well, that's going to lead us to consider something called electron transport and the processes of oxidative phosphorylation. But those are best left for another discussion. Hope you've enjoyed this podcast. Thanks for listening. We'll see you soon.